Hey everyone, I don't know what's going on with my camera there. I seem to have fixed it. Anyway, I'm going to go through regulation of gene expression for you for prokaryotic and eukaryotic organisms. This should be a review of the operon information that you were covering earlier, and then also talk about how eukaryotic organisms go through and regulate with a couple other fancy processes. These must knows are going to be specific for the AP Bio test. So if you are going to be taking that, you can go through the notes and check these out and see, kind of what is expected to be known for that. So we're going to focus on bacteria for this first part and talk about our operons. I know we already kind of did our activity along with that, but bacteria are going to be found everywhere, and they are utilized. Specific bacteria like E. coli as a common organism that is used for um, uh, stealing their genes. I'll just say that because it's really easy to take the genetic material of bacteria since they're very simplistic and it's easy to acquire. Um, and then utilize their control of gene expression for various projects and development of specific genes and organisms, even. So, thinking about our messenger RNA vaccine, um, being able to utilize bacteria's gene expression process, it actually is more efficient. So, you can take the genetic material that you're trying to um, transcribe to messenger RNA, inject it into a bacterial cell like it's its own, and it can go through and replicate really, really quickly. And then it can go through transcription as well through this control, which is kind of cool. Anyway, talking about our operon here. Operon is going to have three pieces to it. It's going to have the promoter, it's going to have the operator, and then it's going to have the specific genes that are going to code for the enzymes within that pathway or the specific molecule that is going to protein molecule that um, is going to help with breakdown of um, whatever you need. So the promoter region up here, again, is going to have the combination of where RNA polymerase can go and attach and do transcription. And it's going to have the operator as well, which is going to act as like an on-off switch, where a repressor or an activator can come and fit into these little teeth and either block the processing of RNA polymerase through, or it can be removed, and then RNA polymerase can go through and do transcription. And then these genes are going to be typically a larger sequence of genes than that. This is a real life one. So our tryptophan operon that's found in bacteria found in our bellies. For when tryptophan is present, it'll go through and do this process. So here's the operon region. Why is this the operon region and over here isn't? Um, Remember, the operon is going to have a promoter, an operator, and genes attached to it. So we have all three over here. Over here on the left side, upstream of our operon, we have a promoter and we have a regulatory gene, but there's no operator. So this would not be the operon region. This is actually called the regulatory gene region, where transcription and translation can occur, and either a repressor or an activator can be made by this regulatory gene upstream from the operator. And in this case, because we're talking about tryptophan, this is going to develop a repressor. Currently, it's inactive. We can see it has its little three teeth here, this orange molecule. And it also seems to have an allosteric site or another site down here where a molecule can bind and kind of change that conformation from being bent to being more linear. And then it can go and travel to this tryptophan operator and lock into place and then repress um, like it should be doing. So we're going to go through that. There's different types, like I just said. There's repressible, where gene expression is active, and then it'll go through, and that regulatory gene can develop a repressor that will then go and attach and block and shut off gene expression, or it could be the the opposite. I don't know what I was thinking there, which could be inducible, where gene expression is turned off. It's not needed at that given time, and then activated by an inducer, or we might call those activators as well, where they attach, where they detach um, and allow for polymerase to be active. So, what does our repressible operon look like? Again, this is where it's gene expression is normally on and active. And this is most typically found in an anabolic reaction where we're creating our macromolecules, because macromolecules are needed all over the place, right? In all different regions of our cell. So this is where they are needed, and gene expression has to occur so that, you know, our proteins can be made consistently and shipped out to where they are. Okay. And then during the repressible operon, that means its regulatory gene can create a repressor, which can then go travel and shut down gene expression once we've achieved the maximum amount of that protein molecule that was being made, and we no longer need it in any given region. But most of the time, we need that molecule readily available. Okay. Um, so we can see down below here this repressor in red. This would be developed by the regulatory gene that's just upstream from this promoter um, operon region. Okay. And currently it's inactive, right? We can see that it's kind of in its bend shape, and then its little allosteric site has nothing attached to it. So it's just kind of free floating. And once that co-repressor, that little molecule to activate our repressor, attaches and locks into place, it changes the shape to flatten out. And then those little teeth can attach and block the processing of RNA polymerase so that transcription cannot occur. So then that shuts the operon off and no longer allows gene expression to occur. So here's an example of that with much greater detail. This is our tryptophan operon. Okay. It's going to go through and be developing enzymes for tryptophan synthesis. So normally, like, let's think about Thanksgiving. That's tryptophan is usually associated to, like, when we eat our turkey and then we pass out at Thanksgiving because we're so tired, right? Normally, we don't have mass amounts of tryptophan in our system. And so we have this tryptophan operon that's creating it um, in order to be used within our system at a normal rate and a normal amount. Okay. Um, and so it's going to be active. And so we can see that transcription is happening here and translation is happening down below, right? So it's creating that tryptophan. Then let's say that we have a Thanksgiving dinner, we have our turkey, and we are now having an excess amount of tryptophan in our system because of what we also digested. The bacterial cells in our gut are going to realize, oh my gosh, there's an overload of tryptophan. We need to no longer make any of it because of this. And so that regulatory gene will be activated. And typically, one of these little subunits down here is going to be that activator, which is kind of cool. So one of those will go through, come and attach as a co-repressor is its name. So it's going to attach to this allosteric site of the inactive repressor, which will then cause it to activate and change shapes. And then it can go through and shut down the process of tryptophan synthesis. Oops, sorry about that. So that no more tryptophan is made because we have a crazy amount of it present. Um, and it can be used all the way up. And then once, you know, all that tryptophan is gone, um, that repressor can detach, sorry, that co-repressor molecule can detach from the repressor, which then causes it to be released and then reactivate the process of our tryptophan operon. So our inducible operons are going to be normally in the off position, and this is going to be most common for our catabolic reactions. So again, when we're taking in and digesting food, right? We don't want to be breaking down um, glucose. We don't want to be breaking down our carbohydrates constantly for that energy release, only when we need it. So there's a lot of different food stores for us on that, right? And so it's going to be in the off position until we need that specific energy. We need those specific monomers of our macromolecules at that time. So it's going to be in the off position. No gene expression is happening. Why? Because there's going to be an active repressor attached to the operator region, blocking that RNA polymerase from going through. And then there's going to be a little inducer, which kind of acts like a ligand, actually, where we talked about ligands before in cell communication, and it'll bind to that repressor region and cause it to go from attached to detached, and it'll pull away and allow gene expression to occur. So it turns that operon on. And so a common inducible operon is for lactose or lactase, which we'll look at next. Again, we don't always have, um, we're not drinking milk constantly. Lactase isn't always present in our gut. But when it is, or when we need it, we'll go through and break it down properly. So there's not that buildup. And so normally it's in that off position until some dairy is digested. Um, and then in this region, that inducer or that ligand molecule, again, down here we can see that being made. It'll attach to our repressor, cause a little conformational change so that the repressor is released from that operator region. RNA polymerase can then go through and create the enzymes for using lactose and the breakdown of lactose, and allow that to occur. And then once um, everything for our lactose has been utilized, then that regulatory gene, RNA polymerase will go through and create that um, repressor again, which will then go and block and shut down that operon. Okay, so our gene regulation can be positive or negative. Um, Negatively controlled. Positive control would be like activated, and we're increasing the amount of whatever that active form is. And then I just wanted to make sure I didn't say that opposite. And then negative control is going to be where we can decrease the amount of something being made. So there's the example of those. I'm going to skip through this cyclic AMP positive control example, but you can look at it. You've already watched that video. So we're going to move on to our eukaryotic gene expression. So only specific sets of genes are going to be expressed at any given time. So we've got around 20%. And it has to deal with the type of cell that we're dealing with, right? Only specific active genes will go through gene expression, not every single gene in every single cell, because that's not effective and efficient for our cells. So we can have some differential gene expression. It's regulated at different stages, so I'm going to point out those areas and then go through that in specifics. So this, again, is for our eukaryotic cells. Chromatin can be modified in some different ways, and the way that DNA is kind of unpacked and unwound is one major difference. We have our RNA processing, so the introns, exons, the cap, and the tail is another way that eukaryotic cells are regulated. And so this is all happening in the nucleus. There's those two spots, chromatin modification and the RNA processing. And then outside, when it goes through translation, basically, there's a couple areas um, for that, which has to deal with the general structure and how um, it's 3D conformation, right? So once our proteins are made and built up, that can be different. And then again, specifically, the protein processing can also be different for gene expression. So if we're going to focus in on the nucleus, we'll start with our first step, how DNA has to condense and go from its spaghetti chromatin structure to actual double helix chromosome structure first, and it has to get unpacked in that way. So how does it do it? There's actually these methyl groups or methylated proteins that will come and attach to our chromatin in our DNA, and then um, that will tighten it up and turn it into chromatin. And then we also have histone acetylation or acetyl groups or acetyl proteins that will attach to the chromatin and cause it to loosen up and relax. So the tighter or the tighter the coils are and how wound up DNA is, the less um, possibility there is for transcription. Sorry, that was weird to say that. And then if DNA is relaxed, then transcription can be performed and it is available for gene expression. So methylation decreases transcription, acetylation increases transcription. So that's the first thing of how tightly packed our DNA unit is. So again, here's a look at that acetylation process. So really tightly packed. DNA can't be accessed right now because of how tightly wound up it is. We have our acetylated histone molecules that are attaching and kind of separating the strands. And so now DNA is loose and transcription can occur. And that's going to be different for all different people out there and eukaryotic organisms. And that's actually called epigenetics, which is something really cool to look into, where we have these slight modifications on our chromatin, and they're little DNA markers basically that are affected, that affect our DNA. And this is based off of, health factors, how healthy you're living, your diet, your exercise, what you're putting into your body. Those are all different markers. Um, and then there's genetic pieces that are passed on from your or from your past generations that are also little markers that attach and cause either methylation or acetylation to occur in different areas. So um, that's one thing you could look into, which is interesting. Anyway, I'm going to skip ahead to our other areas of gene expression modification. So um, the next step, once DNA is unwound and those genes are present, the transcription process, again, we talked about activators versus repressors and how they can increase or decrease transcription. Those are kind of specific transcription factors that will bind to our genetic information and then also cause, a positive or negative control of transcription. So again, we have these regions like we talked about with our operon, and then over here is where those specific elements can bind and either increase or decrease the process of transcription. Okay. The other step is the fact that when RNA is processed, we have our pre-messenger RNA transcript here versus our mature one or our final transcript. We know that exons are going to be linked together and introns are going to be snipped out. And then it kind of depends on those intron sequences that are being snipped out. And that will lead to our final messenger RNA transcript. So sometimes there are little codes that are left in there. or these exons may not be going and may not be bound in this specific order. So this is exon 1, this is exon 2, this is exon 3. That's one way they could bind, or 2, 1, 3, or 3, 2, 1. It really is going to depend on how it gets folded together. So we've got this initiation complex over here where it can be activated or decreased. In this case, if it's activated, we can have some different binding sites and different folding patterns, like I was saying, for transcription to occur. So I'm going to just skip through that. Again, this is that alternative splicing that I was referencing, how introns are going to be removed, and then we can have different sequences of messenger RNA and different orders of those exons. Once that goes away, we can see at the bottom left how that messenger RNA transcript goes 1, 2, 3, 5, and the one on the right goes 1, 2, 4, 5. It all depends on how that gets activated. And then there are little micro RNAs or interfering RNAs that can bind and block translation as well. So once that messenger RNA transcript leaves, right, it can go through and block translation and shut it down, or it can assist in the movement of that messenger RNA complex to the ribosome. So we can see if it does, those little ones attach and bind, it's going to shut down the process and cause that messenger RNA transcript to break down. And so here's that full breakdown of all the different modifications that are attached there.